Starting with the eukaryotic cell. First of all, they are those cells which have a nuclear membrane. That is the first difference we always talk about in a prokaryotic and a eukaryotic cell, that all the eukaryotes, they have a well-defined nuclear membrane and other membrane-bound organelles. Membrane-bound organelles, right? That are present in the cytoplasm. That means there is extensive compartmentalization of cytoplasm. All right, first point and second point, and thirdly we see their genetic material it is arranged in the form of chromosomes, and these chromosomes lie in the nucleus. Clear? When we talk about that they have a complete nuclear membrane, they have membrane-bound organelles which is in the cytoplasm. Obviously, these all are surrounded by a structure which is known as cell membrane, which will not allow any of these membrane-bound organelles to move out or any material to move in and out without permission. So the components, the basic components with which we start the eukaryotic cell is, first is cell membrane, also known as plasma membrane. What is it composed of? It is mainly composed of lipid bilayer and proteins. Then the proteins which are mainly present on the membrane, these membrane proteins, these are classified into, they can be integral and they can be peripheral, depending upon the location, whether they are on the periphery, that means on the wall, on the top, or whether they are integral, in between. Then we see the membrane of human erythrocyte, that means human RBCs, erythrocytes, has 52% of proteins and 40% lipids. That is the composition. Then we see the model. This model of a cell membrane, it is proposed by Singer and Nicholson, and it is a widely accepted model. We call it fluid mosaic model. Fluid mosaic model, and it is proposed by Singer, Singer, and Nicholson in the year 1972. This is the only widely accepted model that we are going to discuss. Okay, let's draw the picture of this model and we discuss what is the basic composition and why it is widely accepted. We see, if we look at the structure of a cell membrane, it somewhat seems like that it has a lipid bilayer. This is the lipid bilayer. Which is present and we know that lipid has a bipolar nature in which the hydrophobic tail and the hydrophilic head is present. Okay, this is a protein molecule. This is the integral protein. That is why it is in between the lipid membrane. Again, this is covered up by the lipid bilayer, one more protein, and a lipid bilayer, if we try to show the three-dimensional structure, we will show so many heads of the lipid membrane onto the upper side. This shows the arrangement of the lipid bilayer, and over here small chains of carbohydrates. This is a carbohydrate chain that has been shown. These are the hydrophilic heads and the hydrophobic tail. This portion is the hydrophobic tail, and this one is hydrophilic head. Phobic, fear from water. Philic, love for water. That's why this head is towards the outer side and the tail is towards the inner side. These are the integral proteins. This is the lipid bilayer and this is sugar. According to this model, we say the quasi-fluid nature of the lipid quasi-fluid, that is 1/4 fluid, liquid nature of the lipid enables the lateral movement, जिसको हम lateral movement भी बोलते हैं, lateral movement of proteins. Within the overall bilayer. Within the overall bilayer of lipid molecules. Then we say the fluid nature. Why is it so important? This fluid nature of membrane. Fluid nature of membrane is important for certain functions. Functions like cell growth, endocytosis, to gulp in something, then formation of intercellular junctions, then cell division, secretion, etc. Okay, these are the functions which are performed by the fluid nature of the membrane. May it be cell growth, may it be endocytosis, may it be formation of intercellular junctions, cell division, secretion. This is basically done by the fluid nature. Then an important property, it is its semi-permeable nature. That means the membrane allows only certain molecules to pass through it. It does not allow all the molecules. It will only allow certain molecules to pass across. Now, moving to the membrane transport. How the membrane transport takes place through this cell membrane structure. Membrane transport, this is one of the most important function of any cell membrane. Why we say most important function? Because it allows the transport of materials in and out of the cell membrane or the cell. It has two methods. One is the active transport. Another one is the passive transport. See, generally there are certain neutral solutes. Neutral means the ones which are normally polarized ions. Many neutral solutes they move across by simple diffusion. They do not need any carrier, they simply move across. Secondly, water moves by osmosis, that we know. Then there are certain other polar molecules which follow facilitated diffusion. Now these polar molecules which need diffusion through a facilitated way, they require a carrier protein. So certain molecules, they require a carrier protein for their facilitated diffusion. And whenever this happens, they pass across using this protein membrane channel. How do they pass across this? They come and fix here, then they move inside, move across, and are released at the other end. This is how they use the carrier protein. And non-polar solvents, they pass across the lipid bilayer easily. And there are few more ions and molecules that are transported against the concentration gradient. And they use whenever any molecule that has to pass across against the concentration gradient, it means it requires energy. That energy is in the form of ATP. This ATP is used, and ultimately this process is known as active transport. How do you have to remember it? You have to remember it, if any process utilizes ATP, ATP stands for active transport. Whenever energy is provided for any molecules to be transported across the membrane. Example, the sodium ions, potassium ions in sodium potassium exchange pump. In this pump, the transport of these ions require energy, and that is active transport. Clear? Now, what is cell wall? Where is it present? Obviously, additional to the membrane, there is a rigid structure present that provides structural strength, is known as cell wall. So we call it is a non-living rigid structure outer to the membrane. Outer to the membrane in case of plant cells, not in animals, but in alga too. So in alga the algal cell wall, it is made up of cellulose, galactans, mannan, and minerals. Which are minerals? Minerals like calcium carbonate, then silicates, etc. This provides toughness to the membrane, that means the wall. And certain other plant cells, other plants, they have a cell wall made up of cellulose, hemicellulose, pectin, as well as proteins, right? Then there is primary and secondary cell wall. You know, there is a junction which is known as plasmodesmata. It connects the cytoplasm of the adjacent cells. And here we write a line to suffice this thing, that primary cell wall and middle lamella are traversed by plasmodesmata. These are the connectors. That means it connects the cytoplasm of adjacent cells. And what is present on the secondary wall? This was the primary wall and the middle lamella. What about the secondary wall? There are pits present. On the secondary wall. Alright. Now moving to a little towards the cytoplasm. Cytoplasm, it shows an array of membranous network. What is that array? It forms a channel. It forms a network of various organelles together, and we call it endomembrane system. Why do we call it endomembrane system? Because the organelles which remain functional here, they are connected to each other as their functions are coordinated. Let's write it down. Some of the membrane organelles, they are considered together as endomembrane system as their functions are coordinated. That's why they are dependent on each other. Each of these is distinct in terms of structure and function, no doubt. Each of these organelles are distinct in structure and function, but yes, their functions are coordinated. That's why they are kept together as a endomembrane system. So which all organelles are included here? First is endoplasmic reticulum. Second is Golgi bodies. Third one is lysosomes. And the fourth one are vacuoles. We know all four. What are these? Where are these located? But how are they coordinated in terms of function? Let's take up one by one. First of all, endoplasmic reticulum. What is it? A network of tiny tubular structures which are scattered in the cytoplasm where? Around the nucleus. So what are the four things that we have to keep in mind? Network of tiny tubular structures scattered in the cytoplasm around the nucleus. But how are they coordinated with the nuclear membrane? They are continuous with the outer membrane of the nucleus. So basically we write they are continuous with the outer membrane of the nucleus. When it joins there, it divides the intracellular space of the two distinct compartments, right? Inner and outer cytoplasm. It forms inner cytoplasm and outer cytoplasm. Means it divides the intracellular space. Intra means within the cell. Intracellular space into luminal compartment. That means within the lumen. This is inner to ER. One is extra luminal. That means same way compartment, which means outer to endoplasmic reticulum. That is, this we call cytoplasm. Okay, so it divides the compartment. This, when bear ribosomes on its surface, we call it rough endoplasmic reticulum and becomes the site of protein synthesis, ER. ER bearing ribosomes is known as rough endoplasmic reticulum, right? And responsible for the synthesis of protein. And when ER without ribosomes, so we call it SCR, and is responsible for lipid and steroid hormones. That is synthesis of lipids and steroid hormones. And when we know about the structure, we draw the structure like this is the nuclear membrane. The outer side, we draw it a bit like this. They may be in the form of stacks, they may be in the form of vesicles. Alright, so this is how these are attached to the outer membrane of the nucleus. And obviously the membrane near the nucleus, it bear the ribosomes. So we show RER here. The dots are the ribosomes. Wherever these would not be present, we call it SCR, and wherever these dots are present, we call that area RER. Clear? Now moving to Golgi bodies. First observed by Camillo Golgi in the year 1898. What are these made up of? Flat, disc-shaped are basically called cisternae. These stack parallel to each other in a concentric manner where near the nucleus. Why? because they are known as packers and movers. Whatever is being synthesized near the nucleus, that is to be packed and moved away, maybe utilized for the cell itself or maybe transported out of the cell. So therefore they are mainly located near the nuclear area. They have a distinct convex cis face and a forming face, and a concave trans or the outer face. I'm drawing it. They have somewhat this kind of structure. Where one end is the forming face, from where the vesicles attach. Ultimately removed from here. Some of it gets concentrated, which later on acts as lysosomes. Now here the forming phase and a maturing phase. This is the forming phase and this is the maturing phase. So this is how the material, whatever is packed, this becomes a part of the complex and ultimately released at the maturing phase. And some of it during the packaging time, some of the material which gets concentrated in the corners may detach later on, becoming the lysosomes. So here they are also known as the packers and the movers of the cell. This is Golgi bodies. Next are the lysosomes, also known as the factories which are going to eat away the cell whenever the cell is in danger, right? They are spherical or irregular, single membrane bound vesicles. This is generally formed during the process of packaging in the Golgi cisternae. These vesicles are formed, or these are formed during the process packaging, during the process of packaging in Golgi cisternae. And what these vesicles contain? These vesicles, these contain a number of hydrolases. Which all hydrolases? Lipase, proteases, then is carbohydrases, etc. Generally, at what pH do they work? They work at a preferably acidic pH, and they show their mode of action. Then the fourth one in this category is vacuole. Vacuoles, we know, are meant as the storage particles. Vacuoles are membrane bound space where in cytoplasm, and the single membrane boundary of a vacuole is called tonoplast. What tonoplast does? Number one, it facilitates transport of number of ions and other molecules. Where? Into the vacuole. When? Against concentration gradient. That means it utilizes energy that is in the form of ATP. Why? Because their concentration remain significantly higher in the vacuole than in the cytoplasm. Whose concentration? Ions and molecules concentration. So again, what this tonoplast does? It facilitates the transport of number of ions and other molecules where into the vacuole. Why? Against the concentration gradient, so that the concentration of these ions and molecules, it remains significantly higher in the vacuole compared to the cytoplasm. It's only to facilitate storage, right? This is about endomembrane system.